Lose Control Rock and Roll Radio, broadcasting live from Hamilton, Ontario, Canada. With my co-hosts, Ben Rispin and Manolis Zontanos. A show dedicated to the purists, the diehards, the ever-beloved fans.
I've never played it.
If you're into the music of the MC5, Alice Cooper, the Dictators, the almighty Blue Oyster Cult, Mott the Hoople, Radio Birdman, Dr. Feelgood, and more, then you must tune in to the award-winning Lose Control. Wednesdays from 3 p.m. to 5 p.m., only on 93.3 CFMU. Tune in locally or hear us online at cfmu.msumcmaster.ca.
Wow, I've never...
I'm wondering why yours always sounded better than ours.
Pardon? I've never played it in my life.
But I was told that I could listen to the radio at a reasonable volume.
There are simply too many notes, that's all. Just cut a few and it'll be perfect. By the powers vested in me by the Federal Communications Commission, I command you to get on the microphone in a serious manner and continue this broadcast.
93.3 CFMU, seriously redefining radio for over 30 years.
This is my boomstick.
Feel overwhelmed when you read research papers? Think research is being conducted in labs far, far away? Well, tune into the AlmaMAC Thursdays from 12 to 12:30 p.m. where we interview McMaster graduate students about their research. You learn about important research that's happening right on campus. Learn about what the guests did before research, how they got involved in academia, and what kind of impact their research can have on you. The AlmaMAC is covering it all from Thursday 12 to 12:30 p.m. on 93.3 CFMU, redefining radio in your community.
Hello, it's Shawn here with you at the AlmaMAC radio, where we reach out and hear from graduate students across McMaster. I myself am a grad student. Well, you know that already. In studio, we have Celeste. Hey, Celeste.
Hey, great to be here.
Yeah, thanks for coming. Yeah, I ran into you a few weeks ago at the SciComm TO conference.
Oh, that was such a great event. My mind is still reeling from how awesome it was.
Yeah, so when I was there, I was like, this human looks very familiar. McMaster, because we met last August at a Welcome Week event.
Yeah.
For the grad students.
Yeah. So it was like, I know this face. It seems very familiar. Oh, wait. There we go. Other McMaster student.
Yeah. Oh, so it was the same for you when you saw me at the conference?
Yes.
Okay, great. Yeah, and now you're here. Thank you for coming.
Oh, my pleasure.
Yeah, so how was the conference for you?
Oh, my goodness. It was such a great experience. It was one of those moments where in the moment while you're there, it's almost as if you can't fully absorb everything that's going on around you. It was so rich with information and folks who were so interested in science communication and doing the best they can to share information. It was a little bit overwhelming, but in a good way, if that makes sense.
Yeah, I felt as though it was too short to get everything in. I just wanted to go to every panel, every workshop, everything, but I couldn't.
Yeah, and picking between concurrent sessions, it was like, wait, do I want to go to this or that? These are both super important. How do I decide?
Yeah, but actually I was happy because Tammy, she did live tweeting for some of the conference, some of the workshops that I couldn't go to. So I felt like I learned a lot from her live tweets, actually.
No, that was super helpful. There was a couple people who were posting up like pictures of slides or recaps, and it's like, thank you, you are lifesavers.
Yeah. But how did you even end up at the conference? How did you get involved in science communication?
Oh, that is a big question.
Okay. You know what? Let's save that for a little later then.
Okay.
So first of all, who is Celeste Suart? What are you studying at McMaster, and how did you end up here?
That's also a big question, but a little bit easier to tackle. So I'm doing my PhD in biochemistry. So I did my undergraduate here at McMaster. So I started off in life sciences, then shifted to biochemistry, thought, okay, this is great. I love the specialization. That was the year the Biomedical Discovery and Commercialization Program was launched.
Yeah.
So then I made the next jump to that.
Oh, nice.
So I changed programs for the first 3 years each time.
Wow.
Ended up staying in BDC because I really loved it. And as part of that program, I ended up doing a 4th-year thesis in the lab I'm in now with Dr. Ray Truant. So he focuses on polyglutamine expansion diseases. So that's a group of diseases. The most common one is Huntington's disease, which most people have heard of if they've gone to a first-year genetics course. It's one of the key examples of autosomal dominant disorders. So I did a 4th-year thesis in his lab. I was thinking, okay, well, I'm going to do Huntington's disease. This is going to be my body of work. And he's like, welcome to the lab. We're going to put you on this project on this disease called SCA1. And I was like, SCA what? So spinocerebellar.
I don't know what that is.
No one does. So it's not just you. It's spinocerebellar ataxia type 1.
This is a rare disease?
It is a rare disease. It's about 1 in 100,000.
Wow. So is it like monogenic?
So it's autosomal dominant, just like Huntington's disease. So they're in the same family of diseases. It has a little bit of different symptoms, though. So it's a form of ataxia, or loss of motor control and balance.
Is that what ataxia is?
Yes.
Okay, great. Thank you.
And most folks out there may have experienced a temporary form of ataxia if they've ever had a little bit too much to drink. So your balance goes, things starts to sway, your speech might be a little bit slurred.
Okay.
But if you've had a bit too much to drink, eventually that goes away. But imagine feeling like that all the time and it not going away.
Wow.
Yeah.
Wow.
Oh, it's not a really great disease. It is awful.
So it's consistent. There are no points where you feel less ataxic. Is that even a term? Ataxia?
Yeah. So ataxic is a term. People are starting to move away from it just a little bit, but it's a progressive disease. So most of the time, folks up until around age 30 or 40 feel completely fine. Nothing seems wrong. And then, one day they might stumble over something or walk into a door. It's like, I'm getting a little bit clumsy. Silly me. But then it gets worse, and it gets worse. And then maybe at work, someone accuses them of drunkenness, even though they haven't had anything to drink.
Yeah.
It's like, something is wrong. I need to get help. And then from that point, it's a slow progression downward.
So do you also get like loss, like memory loss, or like what other symptoms? Like, because if you have too much to drink, this is not my reference note for ataxia.
Yeah.
Some people can black out, or they just wouldn't be able to recall certain events throughout the day or around the time they were drinking. Like, what other symptoms?
Okay, that is really common for people to think like that. At the beginning of disease, when there is an obvious motor phenotype, when people are having trouble with their balance, at that point there is no effect on their cognitive abilities. They're still thinking very, very clear. People can get frustrated, though, as the disease progresses. People will have difficulty with swallowing and speech. So even though their thoughts are moving at the right speed and they have things they want to say, they might have trouble saying it as things progress. Near the very end of the disease, sometimes there is some confusion that comes along with it, but that's usually the very end stage of things.
So what can we do to prevent this? Like, what is your scope of this project? Because this sounds like a really big task.
Very big problem. And luckily, I'm not the only one working on it. I'm the only one in the lab focusing on a SCA1 project, but there's many labs around the world who are trying to come up with ways to tackle this problem. Since it is a monogenic disorder, one mutation, one cause, it's a little bit easier than, let's say, with our colleagues who are studying Alzheimer's disease or Parkinson's disease, because they have very heterogeneous causes and very heterogeneous symptoms. For us, at least, we know, okay, someone definitely has SCA1 or they definitely do not. So we've known about the mutation that causes SCA1 and other polyglutamine disorders like Huntington's disease for about 30, 20 years now.
So still pretty recent. Wow.
So we've had the genetic test, so people can get genetic testing when they turn 18 to see what their gene status is. But we still don't have a very good understanding of what these proteins do in the human body. Early on, there was a lot of emphasis on trying to characterize what goes wrong with disease. There wasn't a whole lot of emphasis on what happens in the wild type just naturally existing in the body. As for Ataxia and for Huntington, the protein in Huntington's disease, these are proteins expressed in every single cell in the human body. And
so in the germline.
yes. And we know that they're required for life. If you knock out these proteins in embryos, they don't develop. So we know they're super important, but we're still trying to figure out what's going on at baseline, just everyday levels.
Right.
So that's kind of where my project comes in. I specifically, I'm trying to characterize what Ataxin 1 does with regards to DNA damage repair.
Okay, so the gene that is mutated is called Ataxin 1?
Yes.
Okay.
And then the protein is also called Ataxin 1.
Great. Okay, so you study this gene and this protein in your lab?
Yes.
Okay.
Because about 4 years ago now, 4 or 5 years ago, there was a group of researchers, really smart researchers, so many so that when the papers were published, instead of putting every single person's name in the author list, they published it under a consortium and then listed names in the supplemental because so many people went into this work. They did what's called a genome-wide association study.
Yes.
So this is where, so it's helpful because we have a monogenic disease, but they're asking, okay, well, all of these folks have, well, the first one was Huntington's disease. All of these folks have Huntington's disease. What other small mutations in the genome could influence when people first start exhibiting these motor symptoms? Because at the time, there was this prevailing hypothesis, which is still around today, of the toxic aggregate hypothesis, that the proteins are aggregating, and because of that, that is where things start to go wrong. So people thought, okay, well, if this is true, then a lot of the genes that will influence age and onset of symptoms will have to do with protein homeostasis, maybe chaperones, protein folding. But what was really unexpected, that came out of completely left field, was the genes that came up of this analysis were involved in DNA damage repair, oxidative stress, and mitochondrial health. It kind of threw the field for a loop.
Yeah.
But it suggested, hey, maybe these proteins have something to do with DNA damage repair. So so they first did it with HD. They then did a follow-up smaller study with SCA1 because there's far fewer SCA1 and other SCA patients than compared to HD.
Huntington's
disease, okay. Yes. I need to, one of the things I'm trying to be conscious of is my use of acronyms. As at this point, I'm so immersed in them.
Yeah.
But when I was first getting into it, I had to write down all of them and have a little cheat sheet at lab meetings.
Yeah. We love acronyms as scientists.
They're everywhere.
Because there's so many words you have to put into a proposal, a grant proposal. You only have a limited amount of space to put all of your thoughts in. So
exactly.
You've got to use those acronyms.
But then if you show it to someone who's not in the field at all, they'll be like, what is this?
HD? Hard drive?
Yeah. Or recently I was talking about I'm going to be talking to a PDF, and I was talking to my mom about talking to this PDF. She's like, are you format? Adobe? It's like, no, postdoctoral fellow.
Yeah.
It's super common.
I feel that. Okay, so are you doing a GWAS, a genome-wide association study?
So that was done previously, and
for SCA1
yes.
in your lab. Okay, cool.
So that was done by another lab. It was grouped mainly in Europe, parts of North America. But when I first came to the lab, it was a really fresh study.
Right.
So my supervisor basically was like, read this, understand it, breathe it, and then you can start doing work. So based on that, we started investigating if these proteins involved in these diseases had any response to DNA damage. And luckily for me, because it's now the basis of my work, they did.
Wow.
Yeah. So we're looking into that now.
Can you tell us the name of the proteins, or is that
No, of course. So the one that I work on is Ataxin 1.
Right.
So what we've been able to show is that when there's a DNA damage event in the cell, Ataxin 1 will actually go to the site of damage and sit down on the DNA. We don't know what exactly it's doing there yet. We are still,
What?
We have this really cool assay or technique where I take cells. It's called a laser stripe assay. So I have these little live cells, unsuspecting. They don't know what's going on. And suddenly I take a 405 nanometer laser and I burn a stripe of DNA right down the nucleus. I kind of I feel like a little bit like of an evil scientist when I'm doing it. Yeah.
What? Okay, where does this DNA come from? what DNA are you?
So it's a bit of a non-specific. We can't tell it's like, oh, it's this specific chromosome.
Okay.
But by burning this stripe in the nucleus, we can say a definite area of DNA has experienced a tremendous amount of non-specific damage.
Right.
And then for Ataxia 1 and also for Huntington's disease, which some of my colleagues are working on, we can then see in like live time, in real time.
No.
And we use fluorescently tagged, I use fluorescently tagged Ataxia 1. We also have some chromobodies, which are like an antibody, but it expresses in the cell live for Huntington.
Wow.
And we can see it as these proteins like, oh, there's damage, gotta go fix it. And they move into the site of the stripe. It is the coolest thing.
So that could actually have applications in cancer as well, because that's what happens in cancer.
Yeah.
You get DNA damage, and some of the genes like BRCA1 or BRCA2 that go to respond to the DNA damage might be mutated. They can't get there. So cell division, cancer. So Ataxia 1, it goes to the site, but we don't know what it does.
Still investigating that. Preliminary evidence suggests that it's going specifically to base damage.
Okay.
Yeah, so instead of like double-stranded breaks or things like that, but this is very preliminary, still investigating it.
Wow.
But it's really neat.
That's exciting.
What's also interesting, and I'm still trying to puzzle it together in my head, is that SCA patients and HD patients are known to have lesser rates of cancer than the general population. Statistically significant, like they don't get cancer as often. So it's like, what does this mean? How are we connecting the dots here?
What?
Yeah.
There's so many questions.
So many questions.
In my mind right now. So, but I would expect differently. Is it that, wait, no, if you have the mutation in the gene, then, okay, what type of,
Yeah.
So this mutation, what type of, like what happens to the protein or the gene? Like, if you have this mutation, does it cause a loss of function? Does it cause a loss of function of the protein in question? Or does it cause like a truncation of the protein? Is it, what happens?
These are very good questions, and there is some debate over loss of function, gain of function within the field.
Well, even gain, yeah.
Yeah, our hypothesis with regards to DNA damage is that this mutation will prevent Ataxia 1 from achieving its goal at the site of damage, whether that is maybe itself. Well, it's not an enzyme. We think it might be a scaffold, maybe. But whatever this polyglutamine expansion, that's what the type of mutation is, whatever that does, it is preventing Ataxia 1 from doing its job. Maybe it's sequestering other key repair factors and preventing them from getting to sites of damage. So it's making these cells worse at DNA repair. So in terms of cancer, that might actually be a good thing, because if you can't repair the damage, the cell will apoptose.
True.
And then dies. Mind blown. Yeah.
True. Wow. Wow.
That is a little bit of hand-waving hypothesis there, but that's the road we're going down right now.
Wow. Okay.
Yeah.
Wow. Okay. Wow. I have a large data set of a bunch of genes, that 10 terabytes of data.
Oh, wow. That's a lot.
Yeah. And yeah, it's quite overwhelming to look at. But I'm going to look for that particular gene in my data set.
Let me know.
See what's happening there. Wow. Okay. So what stage of your PhD are you in? So
I transferred from the master's program last year. So not this past December, but this December before that.
So you're in your second year.
Yes, my timeline's a bit like mixed up. So I'm like year 2.5.
Yeah.
Ish.
Yeah, I feel that.
Yeah.
And how, like, what made you feel that you would do the transfer from master's to PhD? Like, how did you grapple that? Did you always know that you were going to do a PhD, or were you a little apprehensive about it?
Very good question. So when I was considering grad school, and in my fourth year, I was like, okay, well, I really think I like this research thing. My project's going well. I like it. It seems like my thesis is a beginning of something, not an ending of something.
Yeah.
But I wasn't quite sure if I wanted to commit to 4 years. So I thought, okay, it's really common in my department to start as a master's and then transfer. It is very normal. It's even encouraged.
Recommended, yeah.
Yeah, because then, let's say you don't like research, which that happens to people. It's not for everyone. Then you can be like, well, I'm going to get my master's. I can show that I've done a lot of work, and then you can go on to the next stage of your life. So I decided, okay, well, let's do that. I'm pretty sure I like this.
Yeah.
I'm pretty sure I love this. But let's be a little bit cautious. So I decided to go the master's route. And then a year into that, I was still loving it. I was waking up and thinking,
yay, research.
yeah, let's do research. Even in the points where nothing was working and I was in like troubleshooting hell, like just like wading through like so many technical problems. It really energized me. It's like, okay, this is what I want to do. Let's jump in.
Yeah, I actually want to touch on that. I feel like we don't prioritize the time that you spend troubleshooting as much.
My goodness.
I feel like a lot of the times we're just like, yeah, like that wasn't important. Look at this exciting result I have.
Yeah.
But those months, years that you spend trying to troubleshoot this one experiment or parts of different experiments is what causes that big win, right?
Yeah.
I feel like we don't prioritize that enough as scientists.
And definitely, like there's a learning curve when you first start research where it's like, oh, well, this isn't working. Oh, it will work next time. And then next time happens, and next time happens, and it's not working.
Yeah.
Like that laser stripe assay I was telling you about, that took, oh my goodness, I'm getting like flashbacks now, like 6 or 7 months to troubleshoot it to get working. And when it works, it's gorgeous. And you get this rush of like, I am seeing something that no one else has ever seen. But like right now, that assay's offline because we're having laser problems and we're troubleshooting it. And it's like, please come back to life. I need you for data. Yeah. PhDs.
So what is next, though, for you in your research with Ataxia 1 and DNA damage?
So I really want to know what's happening at the site of damage. What other proteins is Ataxia 1 interacting with? We also have some patient-donated fibroblast samples that...
What are fibroblasts? Sorry.
Oh, fibroblasts are skin cells.
Yes.
So we use these cells or get them donated frequently because unlike neurons, which are in the brain, it's a lot easier to get a fibroblast sample from people who are still alive. It's really easy. You can do it's like go into a clinic, get it done, leave the next day. Super non-invasive, or as non-invasive as giving cells can be.
Right. So the cells you work on in the lab, what type of cells are they?
So we use a lot of patient-derived cell lines.
Okay.
So one of my colleagues, actually her thesis was just generating these like Huntington's disease cell lines and characterizing them, and it's such
not easy.
Oh my goodness, the amount of work that went into that is mind-boggling, and it was so well done.
Yeah, that is, as you said, a thesis in itself.
Yeah, it was like that body of work is mind-boggling.
Yeah.
So we have those for Huntington's disease. We have mine for SCA1 that we're starting to develop. We have a few control cell lines. What is interesting about the cell lines we use, since we're interested in DNA damage repair, is that many cell lines, like HEX or HeLAs, are cancer-derived cell lines, which means they're hardy, they'll stick around for a very long time.
They'll proliferate forever.
Yes, but their DNA damage mechanisms and systems are impaired. So when you're trying to study DNA damage,
you shouldn't.
It doesn't work. So we use something called HTERT immortalization.
I read about this in October last year randomly. It's really interesting.
So basically we overexpressed human telomerase, which keeps the telomeres in the cells long forever. So it keeps them alive while preserving their DNA damage mechanisms and responses and their P53 pathways. So we know that the results that we get and the pathways we were studying are likely to be what's actually going on in live cells as opposed to a cancer phenotype.
So how So linking this now to bumping into you at SciComm TO, how did you get involved with science communication?
So this actually ties back to when I was first joining the lab. So when I was trying to figure out what I wanted, what I wanted to do, what lab I wanted to go to for my fourth year thesis, I wasn't quite sure what type of research I wanted to do. I didn't have something that really spoke to me at that point. But I knew that I wanted to be in a lab that had some sort of outreach component that, humanized the science. I had seen sometimes you get so wrapped up in how cool science is that you remember that you forget to remember that this can have an impact on people out there in the real world. And I wanted to be in a lab that had that aspect to it, that had that outreach, which is why I chose the Truant lab. They have really strong connections with the Huntington Society of Canada. And of course, then I was put on the project for SCA1 that doesn't have an outreach component, or didn't at the time. So then, when I was attending my first national meeting, a National Ataxia Foundation meeting, there was a bunch of us trainees. We were talking after one of the sessions, and it's these conferences, they're joint patient researcher conferences. So patients can come up and ask questions about our research.
I love that.
Which is so cool.
I love that.
Yeah.
The last cancer conference I went to was the same thing. Every single session was introduced by a patient
so neat.
who was in some way affected by cancer. And they were able to lead discussions, ask questions, and just hear from what researchers are doing. Researchers then get to interact with these patient advocates. And I find that to be so important because scientists, don't get me started on the persona of science. Generally, we're just so far removed.
We're in our ivory tower. And we'll sprinkle knowledge down every once in a while. That doesn't work.
No.
We need to...
No.
Yeah.
Sorry.
No
Your
worries.
Communication.
So at this conference, afterwards, we were discussing, it's so great that the patients are here and they can listen to our research and we can share with them and they'll give us ideas. It's too bad this only happens every 2 years.
Yeah.
I wish there was a way for them to find out more. And then this came this idea. It's why don't we make something where they could access the research online? So Huntington's disease already has, or at the time already had, this knowledge translation platform where Huntington's disease research was transformed into lay summaries. And a couple of us got together for SCA1. We're let's do it for our patients as well. So about a year and a half ago, we launched SCAsource.
Wow,
which is
I like that.
Yeah, I'm very proud of the name. So it's an online platform where research on spinocerebellar ataxias, and now due to interest, we're expanding to other forms of ataxia as well, is research on that, as well as varying topics that are tangentially related, is translated into plain language summaries that hopefully everyone can understand.
That's amazing.
Yeah.
Yeah, I've known of these types of resources for other rare diseases. It's blanking on me now, but there's this database of patients who actually started their own trial.
Yeah.
I can't remember what
I know what you're talking about, and I'm also blanking.
But yeah, they started their own trial because they all got together. They had all of the research that was there from the researchers, and they started their own research. That is amazing. SciComm is great.
Phenomenal.
So, and there's a conference, a workshop happening.
Yes.
ComSciCon.
Yeah.
GTA.
Yeah.
So that's happening. If you're interested, you're listening and you're interested, I'm sure you can find it on the Google. I saw it on Twitter.
Yes, it's big on Twitter. I think applications to that open the end of this week. Be a really great opportunity.
And it's a 2-day workshop, I think, over a weekend. So, yeah. Sign up if you're interested and you're listening. So we don't have much time left. Is there anything else you want to add about your research, in these last 30 seconds?
Just to let people know, go out, get involved in SciComm. It is super rewarding and really, really important.
And where can we find you? So if there may be anyone listening that might want to contribute cells, fibroblasts, or anything.
Yeah, reach out to me on Twitter. I'm at Suart CE, so SUARTCE on Twitter. We also have a lab website. If you Google Truant Lab, I'll come up.
Yeah, so we'll put it in the show notes, so you can log on to our website, which we have, yay! And you'll see the links there for Celeste. So thank you so much, Celeste, for joining
Well, thank you for inviting me.
on the AlmaMAC, and we'll keep in touch. I want to hear more about your research. Yeah. So thank you for joining us on the AlmaMAC. Stay tuned for Get Lit with Jamie.